So data integration and or batch correction. First, we revert back to the raw data, and then we set the log transformation to none, because in some version of AND data there are some issues, so it's just technicality. And then we should also make sure that whether the data is normalized or not. So the way that we do it is just take the given set of values, and when it is normalized we can see that the values are in decimals. Now that we have the normalized values, we can extract the highly variable genes that we detected earlier, and then we see that there are 2,621 highly variable genes. Now we extract variable genes in each sample. The way that we do it, we set the batch key in the highly variable function to sample, and then we generate some stats here, and then we count how many cells or how many genes we have in given batches, and then we get a list, and the list looks like this. So we have 889 genes, showing no variation whatsoever in any of the samples. On the other hand, we have 122 genes that are highly variable across all the genes on all the cells. And the recommendation is to focus on the genes that are variable in at least given number of samples or batches within a set. And here we set it to 2, and then we can see that there are 4,270 genes which are highly variable. Now let's look at the approaches that we can use for data integration: Harmony, BBKNN, and Combat. Just for interest of time, I will walk you through Harmony, but and I skip BBKNN and Combat, but we will make a comparison between them, because you could see how they perform. It's also important when it comes to assessment of the data integration. So you can run different approaches, different methods, and then eventually check what will be the outcome. So we can first check what we have in data. So we have the observation, var slot, unstructured, which you have in general information, which isn't associated with any of the in terms of structure and data. I can show it to use unstructured, or like the like you can think of like a metadata that you have, and given set of parameters that is not possible to link to any of the other variable that we have in our data set. Then we have the ops-m, which has the PCA. It is for the dimensionality information, and then observe multi-dimensional, if I remember correctly. We have then the variables like principal components, variable multidimensional, and then some extra information like connectiveness and distances. Now, let's run the Harmony with this Harmony integrated, and then we set 2 samples. We pass the PCA Harmony as the main XPCA, and then we calculate neighborhood, UMAP, and then we do the clustering with Leiden. I will talk about clustering later. Then let's first run this chunk, and then we do the visualization both for the corrected and also in Harmony corrected UMAP. And then we save the data. So here is the output of Harmony integration, and then this is the first picture that we have from UMAP before data integration. So we can see that some of the discrepancies that we have, I mean in terms of separation of the samples in the space with potential batch effect disappear here. We see a better overlapping between the among the cells in the UMAP space. Now I jump, and then I would like to have a bit more discussion when we are comparing the different approaches. So we have BBKNN, we have combat, UMAP harmony, and then uncorrected data set, which is the original picture that we have. So just to recap, the goal of integration is to align cells with similar expression profile across batches or samples, while at the same time we are trying to preserve the biological differences. Again, in the uncorrected data, we could see a clear batch effect, some samples forming distinct clusters. Combat and BBKNN reduce this to some extent, but we still see batch-associated separation. In particular for combat struggles a bit more here. You can see some of the samples, cells from some of the samples are still separated in the space. And in BBKNN, we don't see that as significantly. Here we have, for example, control 19, but if you look at the harmony, we see a better alignment across samples while preserve the distinct cell population. So all in all, for single-cell RNA-seq, non-linear integration approaches like harmony are preferred compared to linear approaches such as combat and Lima. I haven't shown Lima, but Lima shows more or less the same picture as combat. Now let's talk about clustering.